Okay, good morning, everybody. I just wanted to talk about a couple of things. First of all, the exam 3, the score is already came out. I think you guys did actually pretty good. Good job, and I also posted this score distribution, the Moodles. And second thing, it's about final exam. I already sent out an email to you guys, and we have an alternative final exam on the December 17th, right, from 9:30 to 11:30, also the same classroom. So I think everybody, if you wish, you can come. You don't need to tell us, you take alternative or the regular final day, right? And the third thing, I think Benji, did I pronounce your name correct? Yeah. Okay. So you emailed me, ask about the bonus point, right? So we have like 3 bonus point list on the new Moodles. Usually what we did usually put like a bonus question that was 3 points in either quiz 5 or quiz 6. So then, so you get a chance to earn 3 points bonus, right? And for the coverage of a final, I also wrote an email that's 7% from the new materials, like fuel utilization exercise, and the glycemic index and diet effect on the metabolism, right? And 30% from the older materials only include the carbohydrate and lipid metabolism, right? And the other like digestion, absorption, amino acids, entire section from Carrie Ehrman, and protein synthesis and degradation, and essential fatty acids are not included, right? Is that clear? Okay. All right, any questions about the final exams?
So lipids are covered, just not essential fatty acids?
Yeah. Lipid except the essential fatty acids, right? Okay, so let's talk about the fuel utilization today. So the main concept in this session that you guys need to know, like we're going to talk about the differences among tissues in utilization of fuels during the fasting or fat fasting condition. Means that different tissues may utilize different fuels and in different physiological conditions. And second things that we're going to learn is called branching points in metabolism. Several major or most important metabolites in a different metabolic pathway. This is called a branching points that is very important to connect different metabolic pathways, right? And then a tissue-specific fuel metabolism. The fuel metabolized is specific in different tissues. Just one example, like ketone bodies, synthesized in the liver, but utilized in other tissues like brain, skeletal muscle, and other tissues, right? And liver does not utilize ketone bodies. They always have a reason, either because of they don't have an enzyme to metabolize energy fuels, and some other tissues they utilize the ketone body, but they can't synthesize ketone bodies, right? It's also because the lack of the enzyme for synthesis of ketone bodies. And lastly, I wanted to talk about the adipose tissue in fuel metabolism. The reason because adipose tissue is kind of a specific tissue that recently has been made, progress to understand the role of adipose tissue in metabolism, and also because my group actually studied adipose tissue, right, in metabolism. So adipose tissue is not what we understand only stored triglyceride as energy, but adipose tissue has also very important metabolic function by releasing the important hormones, and peptides, and regulating the whole body metabolism, right? Okay, now metabolic fuels. So definition for metabolic fuels. What is a metabolic fuels? What kind of molecules are considered metabolic fuels? So the definition for metabolic fuels is that circulating compounds that can be taken up by the tissue or cells for energy production, right? So the two things that you need to remember for metabolic fuels. First is the compound has to be present in the circulation, right? The circulating compound, that means it can be picked up by any type of tissues. And then the second thing, that these molecules can be metabolized for ATP production, right? Circulating compounds and can be picked up by tissues and generating ATPs, right? So this kind of compound you consider the metabolic fuels, right? And what are the major metabolic fuels that we have learned or we know? Can you guys name a number of metabolic fuels? Yes, glucose and fatty acids, right? I heard fatty acids, yes. We have 3 more metabolic fuels that we have learned. Ketones, yes. Oh, sorry. Triglyceride, right? Ketones and what's that? Yes, amino acids. Ketone bodies, amino acids, right? So all these molecules, they can be released into the circulation and picked up by other tissues and oxidized in the cells and producing ATPs, right? And one important concept you guys need to know, not all these metabolic fuels are available at the same time. That means in the fat conditions, ketone bodies will not be available, right, for energy, because we only need to use ketone bodies during the fasting. So in other words, that ketone bodies only are available during the fasting for energy, for metabolic fuels. And 2 sources of metabolic fuels we already talked about a lot, right? Endogenous or exogenous. Exogenous means that energy fuels are coming from diet, right? So dietary fuels we consider sort of exogenous energy fuels, right? So dietary glucose, fatty acids, amino acids, right? Exogenous. So exogenous fuels are only available during the feeding condition, right? Fat condition. So we utilize energies that we absorbed from diet. And the other sources of energy fuels or metabolic fuels from endogenous. So our body either store or can synthesize or can make the energy fuels, right, during the fasting condition. So endogenous fuels, metabolic fuels, our body use during the fasting condition. The first category of this endogenous fuel is storage, the stored energy fuels, right? Do you guys still remember the storage energy fuels? What are the triglyceride, right? And glycogen, triglyceride, glycogen, right? And one more energy fuels that also can be stored in the body, and we can use it during the fasting. Protein, right? Only under very long term starvation condition, our body start break down proteins and utilize as an energy fuel, right? And in order to the stored energy fuels, and our body can make also, right, energy fuels. And two important energy fuels that our body can make. What are these? Ketone bodies, right? And most important thing is our body can make. We always wanted to keep blood glucose level at a normal range, right? So glucose, right? Through gluconeogenesis. So glucose and the ketone bodies are the two important metabolic fuels that our body can make during the starvation. So glucose made from non-sugar precursors through the gluconeogenesis pathways, and ketone bodies made from acetyl-CoA, right? All right. So during the fat condition or the feeding condition, what are the major metabolic fuel, energy fuels that our body use? Glucose. Thank you. Glucose, right? During fat condition, and this glucose mainly from diet, right? Dietary carbohydrate. And when food is not adequate during the starvation, our body adapt to utilize the sort of endogenous fuels, right? To keep the most important thing, to keep the blood glucose level at a normal range, because the brain and other like red blood cells only rely on, or heavily or only rely on the glucose for energy. So it's very important. We either to break down the stored glycogen to maintain blood glucose level, or we made glucose through gluconeogenesis, right? And of course, in very long term starvation, we also make ketone bodies. So here just wanted to talk a little bit more about the fasting condition, how we keep the balance, utilize storage, the stored energy versus utilize stored energy, right? So in the fat condition, we have excess energy, so we need to store energy just for the use during the starvation. So the first thing is to make glycogen from glucose, from dietary glucose, make glycogen and store in the liver and the skeletal muscle, right? And a second important thing is to store triglyceride primarily in the adipose tissue. And fasting condition. So fasting condition that we already know that body utilizes store energy and synthesize fuels, right? So first, we need to break down glycogen to release glucose, right, from the liver and not from the muscle, right? You guys know. Muscle will not release glucose because of lack of glucose, 6-phosphatase, right? So this is the most important thing. And as the starvation continues, then liver and kidney will start to make glucose through the gluconeogenesis. And also, bodies break down the triglyceride and utilize triglyceride, right? Release fatty acids and pick up by other tissues like muscle, and of course the other tissues can also use fatty acids for energy. And as starvation prolonged, takes longer, then we need to synthesize ketone bodies and release ketone bodies. And we also mentioned earlier the reason why we need to make ketone bodies, although we have enough fatty acids, right, from triglyceride breakdown, because our brain cannot use fatty acids. So this is one of the important reasons for our body to make ketone bodies to allow the brain to be able to use the energy fuel. All right, so the main purpose during the starvation, our body is try to spare glucose and amino acids, right? So that means we utilize a lot of fatty acids, fat, and also make the ketone bodies and make glucose, right, to spare the glucose and amino acid. Okay, here's something about the transition between 3 macronutrients, the carbohydrate, protein, and fat. So first question is, can protein be converted to carbohydrate? Yes. How? Okay. Yes, right? So protein can be converted to carbohydrate, can be converted to glucose, particularly during the fasting condition, right? When muscle break down a lot of protein, release amino acids, and we know a lot of amino acids is glucogenic, right? Through the gluconeogenesis, right? So a lot of amino acids are the precursors of gluconeogenesis. So, and this process occurs during the fasting, right? Protein can be converted to carbohydrate to maintain the blood glucose level. And what about the carbohydrate converted to fat? Yes, right? Through the lipogenesis, and this process occurred in which physiologic condition? Feeding condition, right? When we have excess the glucose, the carbohydrate from the diet, and will be converted to fat through the process or pathway called de novo lipogenesis, right? And protein to fat. Yes or no? Yes. How protein being converted to fat, like fatty acids? Through the which pathway? Remember? The same pathway, right? Lipogenesis. Protein be oxidized to acetyl-CoA and provides substrate, right, for fatty acid synthesis. Right? So, and this pathway is also occurred in the fat condition, right? In the fat feeding condition. Okay, now opposite condition. That's fat, or is fat able to convert it to protein or carbohydrate? Yes or no? No? Oh, sorry, the first question actually, carbohydrate to protein, right? It's not be able to convert a protein, right? And fat, it basically is not. But some specific fat actually be able to convert it to carbohydrate. Fat is not able to convert it to protein, but specific type of fat, right, is able to convert it to glucose. For example, propanyl-CoA and glycerol are also gluconeogenic, right? The precursors of glucose, neogenesis. So now let's talk about the specified or specific type of fat and how they are converted to the fat to glucose, right? So first one is the glycerol. So glycerol is generated from the hydrolysis of triglyceride, right, during the fasting. And glycerol is released from adipose tissue, then liver can pick up for gluconeogenesis, right? And it's sort of the pathway for gluconeogenesis starts with the glycerol. Remember, when we talk about the gluconeogenesis, and this is one pathway that only occur in the cytosol, and it just it takes like two reactions, right? Remember the two bypass reactions. And this first is a glycerol kinase. You can convert glycerol to glycerol 3-phosphate, and this being able to convert it to glyceraldehyde 3-phosphate by this enzyme glycerol 3-phosphate dehydrogenase, right? And this will enter the gluconeogenic pathway. And the second type of specific fat that can be converted to carbohydrate is propanyl-CoA. Propanyl-CoA basically is produced from two pathways, right? One is alt-chain fatty acid oxidation, and branching amino acid metabolism, right? The two pathways, alt-chain fatty acid oxidation and branching amino acid metabolism, can all generate propanyl-CoA, right? And this again is the pathways that how propanyl-CoA being converted to glucose. First reaction is being conversion of the propanyl-CoA to methylmalonyl-CoA, and this enzyme propanyl-CoA carboxylase is enzyme to catalyze this conversion. And the next step is the methylmalonyl-CoA mutase, right, to convert methylmalonyl-CoA to succinyl-CoA. Succinyl-CoA is one of the intermediates in the TCA cycles, remember, and then utilize some of the pathways, the TCA cycles, to convert it oxaloacetate, which then leaves the TCA cycles and enters the gluconeogenic pathway for glucose production, right? So this is the the process, how propanyl-CoA can be utilized for glucose production, for gluconeogenesis, right? Again, the two specific type of the fat, glycerol and propanyl-CoA, can be gluconeogenic, right? And the primary CoA basically from the two sources. One is from the alt-chain fatty acid oxidation and branched-chain amino acid metabolism, right? Any questions about this process? So now let's talk about the branching point in metabolism. So branching point is the substrate that can be connected in the different metabolic pathways. And they are very important intermediates in the metabolism. There are actually 4 of them: glycerol 3-phosphate, pyruvate, acetyl-CoA, and succinyl-CoA. Right? So now let's talk about the glucose 6-phosphate. Try to remember which pathways that can generate the glucose 6-phosphate.
The liver can phosphorylate it.
Yeah, so glycolysis, right? Glycolysis, this is the first reactions. Glucose can be phosphorylated to glucose 6-phosphate. And glycogen breakdown, glycogen lysis. And gluconeogenesis, right? So these are the 3 pathways that can produce the glucose 6-phosphate. And the pathway that can use the glucose 6-phosphate, let's see, is glycogen synthesis, right? So glycogen can be synthesized from glucose, and glucose 6-phosphate is one of the intermediates in glycogen synthesis. And glycolysis and pentophosphate pathways, right? Remember, the substrate is glucose 6-phosphate, and for the specific pathways. On a second branching point that is important, metabolize the pyruvate. So pyruvate can be produced from glucose and other gluconeogenic amino acids, right? Yeah, this diagram I think probably is better, more clear about the the pyruvate and connect the different metabolic pathways, right? For example, pyruvate can be generated from glucose metabolism glycolysis pathway, right? And lactate and alanines and other amino acids can also be metabolized to pyruvate. And pyruvate can be used or metabolized through the different pathways. For example, can be further oxidized to acetyl-CoA and either enter the TCA cycle for oxidation for energy production, or acetyl-CoA is a very important substrate for all these fatty acids, ketone bodies, and cholesterol synthesis, right? And pyruvate can also be directly converted to oxaloacetate into the gluconeogenic pathways, right? So this just help you to connect all these different metabolic pathways, right? And they actually interrelated, right? We mentioned earlier. So pyruvate is very important substrate that connected all different, glucose and fatty acids and amino acids metabolism, right? And of course, these different pathways depend on the physiologic condition, right? For example, in the fat condition, feeding condition, pyruvate most likely to go acetyl-CoA and TC cycle for complete oxidation, right? And also for the fatty acid synthesis and cholesterol synthesis. But during the fasting condition, the pyruvate mostly go to the acetyl-CoA and for ketone body synthesis, right? And gluconeogenesis. Acetyl-CoA. So acetyl-CoA is, of course, is very important central molecules, right? And can be produced from the oxidation of three macronutrients, fatty acids, glucose, and amino acids. And it also very important molecules serve as the substrate for fatty acids, ketone bodies, and cholesterol synthesis, right? We just talked about in the last slide. And of course, it can also be enter the TC cycle for energy production. And succinyl-CoA. Succinyl-CoA actually connect the TC cycles, right, between the branching amino acids and out-chain fatty acids metabolism, right? Remember the succinyl-CoA basically is generated from the oxidation of a branching amino acids and out-chain fatty acids. And this enter TC cycles, right, can either be used for the gluconeogenesis during the fasting condition. And this slide, I will just put all the four key points or key metabolites together and connect all different metabolic pathways that we learned, right, in this semester. So you just try to know that where are these four important substrates located and how they connect to different metabolic pathways, right? So glucose 6-phosphate, pyruvate, acetyl-CoA, and succinyl-CoA here in the TC cycles. We know glucose 6-phosphate can be generated from glucose oxidation, from glycogen breakdown pathways, and also gluconeogenesis pathway, right? And it also connect and can be used for pentose phosphate pathway, right, for synthesis of a 5-sugar carbon phosphate. And it also can be utilized for the glycogen synthesis in the fat condition. And of course, it can be further metabolized, right, and oxidized through this pathway. And pyruvate, we know that can be generated from glucose metabolism and amino acids metabolism. And it can be either further metabolized in the feeding condition to acetyl-CoA and completely oxidized in the TC cycle, or it can be used for gluconeogenesis during the starvation, right, conditions. And acetyl-CoA molecules can be the product of the 3 nutrients oxidation, right? Can be oxidized from, can be further oxidized in the TC cycle in a feeding condition, or for fatty acids synthesis, ketone body synthesis, and cholesterol synthesis, right? And succinyl-CoA is a little bit simple, and the only is a metabolite of a branching amino acids and out-chain fatty acids, right? and it's important substrate for gluconeogenesis in the TC cycles. Right? Any questions? Okay, now let's start talk about the specific tissue-specific metabolism of fuels. So meaning that in the different tissues, they utilize different metabolic fuels, right? They have a different phase. Metabolic fuels have different phase in different tissues. So let's begin with the brain. So the metabolic fuels that brain can utilize for energy, of course, most important energy fuels, glucose, right, for brain. And brain also can utilize ketone bodies and lactate. Because the brain lacks of energy fuels, as energy fuel storage, right? The brain usually don't store any energy fuel in glycogen or triglyceride. So it requires energy constantly, the continuous supply of glucose or ketone bodies. So in the feeding conditions, about 120 grams of glucose being utilized per day by the brain, right? It accounts like 50% of the glucose that utilized in the body, right? Sorry, the 60%. 60% of the glucose that utilized in the body, of total, glucose actually utilized by the brain. But you guys also probably wanted to know that in addition to take up energy fuel to utilize for ATP production, the tissue can also contribute energy fuels to the circulation and used by other tissues. So you probably wanted to know what kind of energy fuel that specific tissue can release, right? And the brain sometimes can also release lactate, particularly during the prolonged starvation period, because even brain utilize glucose, and during the very long starvation, prolonged starvation, then brain only partial oxidize, right, and to produce the lactate. So the lactate, so brain also can release lactate, although the amount probably is relatively small compared to the skeletal muscle and red blood cells, but they also release some of lactate. So lactate can be picked up again by the liver or kidney, then the made glucose, and then release again. So kind of recycle, the glucose, right? And during prolonged starvation, so in the fat condition, of course, the brain mostly utilize glucose as energy fuel. But during the prolonged starvation, the ketone bodies can be oxidized, right, and probably even utilize more ketone bodies compared to glucose during the prolonged starvation. Again, the purpose of this is also try to spare glucose, right? And glucose metabolism is restricted to the glycolysis and stop at like lactate production, right? During the prolonged starvation. Again, so brain in the feeding condition primarily use glucose as energy, and during the starvation, particularly prolonged starvation, brain is able to utilize ketone bodies. And for the glucose metabolism during the, again, prolonged starvation, brain just does the glycolysis, right, and produce lactate. So lactate can be released from the brain during the prolonged starvation, right? Is that clear? So brain is pretty kind of simple. Um, so here's a question. What is the brain's preferred fuel? Glucose, right, of course, and heavily rely on glucose and ketone bodies and lactate sometimes, and only under the very specific condition or particularly pathologic conditions like hyperglycemia, when very, very low blood glucose level on these conditions, the brain can also pick up some lactate and utilize as energy, right? Okay, so let's take about 10 minutes break. All right. Okay, let's get going. Um, so now, skeletal muscle. The fields that can be utilized in the skeletal muscles, almost all type of the metabolic fuels can be used or can be oxidized in the skeletal muscle, including glucose, fatty acids, including either stored or circulating. I just remind you that skeletal muscle is also stores some amount of a triglyceride, right? So skeletal muscle can utilize the stored triglyceride as well as the circulating fatty acids that initially comes from adipose tissue, right? And ketone bodies, amino acids, right? So what is the preferred fuel in the fat or under burst of activities? So it's quite different, right? So basically, it's the glucose, right? Glucose is a preferred fuel for skeletal muscle during fat condition or during exercise. And what is a major fuel in the resting muscles or in the fasting condition? We don't consider exercise. During the fasting condition, what are the major energy fuels for skeletal muscle? Do you guys know? Fatty acids, yes. Sorry. Okay, so fatty acids needs about 85% of energy needs, right, during the fasting condition. An important thing is what kind of metabolic fuels that muscle can release. That means that the muscle can contribute to the metabolic fuels or can make metabolic fuels and release into circulation. That includes lactate, alanine, and glutamine, right? So remember the lactate and alanine are the precursors of gluconeogenesis, right? Remember the two important cycles in the gluconeogenesis? Cori cycle, right? So lactate is used for Cori cycle, will make glucose in the liver, and muscle is a major contributor to the lactate. And alanine is a alanine cycle, right? Remember, for the gluconeogenesis. And muscle can also release a glutamine. Yeah, here's a cycle. Just want to remind you again, the Cori cycles, the muscle as a major contributor to the circulating lactate together with the red blood cells. So lactate being picked up by liver and make the glucose, right, and released to the circulation and used by other tissues, including muscle and red blood cell. And alanine cycles, right? So muscle release alanine and transport it to the liver and do the so-called deamination, again converted alanine to pyruvate and for gluconeogenesis in the liver, right? So muscle itself utilizes glucose, particularly mainly glucose in the fat condition or during exercise, and utilize the fatty acids and ketone bodies in the very late stage of starvation, right? And also amino acids for energies. And during the fasting, fatty acids are the primary energy fuels for skeletal muscle. Right? It meets like 85% of the energy used in the skeletal muscle during the fasting. And muscle contribute to or release lactate, alanine, and glutamine. And alanine, lactate, and it's very important. Again, the gluconeogenic precursors through the choline cycles, alanine cycles, right? And storage energies. So muscle can store energies. You guys remember which stored energies the muscle primary has? Storage and stored energies. Glycogen, yes, glycogen is the primary, right? The major stored energy in the muscle. And what are the other stored energy that we just talked about it? Triglyceride, right? So triglyceride can also be stored in the skeletal muscle. So during the contraction, the muscle contraction, we're going to talk about this later, during the exercise session. But during the exercise, glucose can be actually partially oxidized to produce a lot of lactate, right? And the lactate production significantly affects the exercise performance, and depend on which muscle fibers, and also depend on the chain and unchained individuals. The production, the amount of lactate production is quite different. And the muscle can also break down the yeah, we need some music. So and muscle break down the protein, right? And use the amino acids during the exercise. All right. Some kind of comments, and you guys already know, right? The glycogen stores and are only used locally, means that the glycogen breakdown for energy production, and glucose will not be released, right? In other words, that muscle does not contribute glucose to the circulation. And again, the reason is lack of the enzymes, right? It's glucose 6-phosphatase. And how does muscle contribute to circulating glucose? Indirectly, if not through the breakdown glycogen, but muscle actually make a very important contributions to the circulating glucose. Remember how? Indirectly. Thinking about it, what kind of metabolic fuels muscle released, and how this fuels being converted to glucose, right? Yeah, alanine and lactate through the choline cycles, alanine cycles. So muscle contributed to circulating glucose through these two gluconeogenic pathways, right? By releasing lactate and alanine, and for gluconeogenesis in the liver, right? So indirectly contribute to the glucose. And the muscle also release the glutamine, right? And glutamine can be converted glutamate by glutamate, glutaminase, and it also can be converted, glutamate can convert alpha ketoglutarate, right, by the called glutamate dehydrogenase. And remember, some of the glutamate is also the important, the energy fuse in small intestine, right? So the muscle release this glutamine and pick up by the small intestine and utilize as energy in the small intestine. So here sort of summarize the metabolic fuel utilization in the skeletal muscle. So in the fat condition, muscle primarily use glucose almost for any type of tissue, right? In the fat condition, always use glucose as a primary fuel. And in the fasting, under resting condition, not considering exercise, the fatty acids are the major metabolic fuels for skeletal muscle during the starvation. And activities, the exercise, muscle also utilize primarily glucose as the energy fuels, right? But under the prolonged starvation, so muscle can utilize all type of energy fuels, right? But the fatty acids, the ketone bodies are the most important energy fuels that muscle utilize. And for glucose, the muscle only oxidize partially, right, and release lactate. And amino acids, again, is only, sorry, I think it's missing kind of part. I don't know, I couldn't find it. So amino acids is also being utilized and generate the protein, actually, the breakdown, right, and produce a lot of alanine, and alanine also released from the skeletal muscle. Kidneys. So kidneys can also utilize almost all type of energy fuels, including glucose, fatty acids, ketone bodies, lactate, and glutamine, right? And how does the kidney utilize this fuels for energy? How kidney metabolize the energy fuels? So basically, we can go almost all the different metabolic oxidation pathways. Like glucose can be either oxidized through glycolysis, TCA cycle, complete oxidized, and fatty acids can be oxidized through beta oxidation, ketone bodies, and oxidized through TCA cycles, right? And lactate, the TCA cycles, and glutamine. So kidney seems to oxidize all this metabolic fuels completely, right? For energies. And of course, the ketone body fatty acids are primarily used during the starvation condition, right? Similar to the other tissues. And kidney, we know as very important organs, right? And they can make a glucose during starvation. So kidney contribute or release glucose, contribute glucose to the glucose, circulating glucose. And the difference is that kidney has this glucose 6 phosphatase, so glucose can be made and released into the circulation. And small intestine. So small intestine is a little bit kind of special. They utilize the amino acids, the glutamine and glutamate, as the energy fuels, and they also utilize glucose, fatty acid, ketone bodies, right? And glucose is also partially oxidized to release lactate from the small intestine. Of course, they compare to muscle and red blood cells, and this amount of lactate released from small intestine is relatively small, right? But they actually able to release lactate because they partially oxidized glucose through the glycolysis pathway, right? And glutamine is also be partially oxidized and to form lactate, pyruvate, and alanine, right? So in general, the small intestine is able to release lactate, pyruvate, and alanine into circulation, right? But it's not like a major contributors, right? Again, compared to the skeletal muscle and red blood cells. And colon. So colon actually is a very specific, unique metabolism, and most of the energies actually it comes from. Do you guys know? No. Butyrate, right? So the most important energy fuels for colon is butyrate. The butyrate is basically from the fermentation of dietary fibers, right? Dietary fibers fermentate in the GI tract and produce the lot of butyrate and short chain amino acids. Short chain fatty acids. So butyrate again is a major energy fuels for the colon, right? Red blood cells, I think this probably simplest cells, right, in terms of energy fuel utilization. The heavily rely on or only rely on the glucose for energies, right, red blood cells. And the reason is red blood cells lacks mitochondria, right? So it cannot utilize the other energy fuels, for example, fatty acids, ketone bodies, and amino acids. For complete oxidize all these energy fuels requires mitochondria, right? The ketone body is also oxidized, right? To acetyl-CoA and TCA cycle, they all occur in the mitochondria. And so again, they only utilize the glucose for energy. And this you guys all know, right? Which other pathways are red blood cells able to perform? Is pentose phosphate pathway, right? So again, just remind you again, in the red blood cells, glucose only is metabolized through the glycolytic pathway and produces lactate, right? So that's why the red blood cells are one of the major contributors to the lactate, right? Circulating lactate. Liver. So in the fat conditions, 20% of blood glucose level, blood glucose actually is taken up by the liver. When we eat after a meal, then we take the glucose from the diet and into the circulation, then 20% of the glucose that initially from diet actually picked up by the liver and metabolized in the liver, right? We think we talked about this before, right? So what liver is going to do is this 20% of circulating blood glucose. First of all, it's of course for the glycogen synthesis in the fat condition, right? And secondly, for fatty acid synthesis, if we have too much glucose from the diet, right, being converted to fatty acids through the de novo lipogenesis. And glucose can also be used for PPE pathways, and the stored glucose, the glycogen and fatty acids, right, like skeletal muscle. And in the fasting condition, the liver is the main sources of endogenous energy fuels. So what type of energy fuels that liver can release during the starvation to provide energy fuels for other tissues use? Can you guys tell? Yes, glucose and during the starvation, including long-term starvation. Yes, ketone bodies. Ketone bodies made by, through gluconeogenesis. Ketone bodies, right? So release glucose and contribute the glucose from the liver contribute glucose actually involve the two pathways, right? Glycogen breakdown and the gluconeogenesis. And also make ketone bodies, right? Right? And in terms of utilization of liver actually does a lot of things, right? And really make very important contribution to the whole body metabolism. But this all requires energy, right? So how liver utilize the energy to make all these, things or does this jobs. So feel that liver utilized includes amino acids, glucose, fatty acids. A little bit surprised, right? Glucose is not the first place. So the preferred fuel that can be utilized by the liver is the amino acids metabolized, like alpha keto acid, right? So alpha keto acid is the main energy fuels that are utilized by the liver for doing a lot of things. And second thing important is that for all this nutrient metabolism, in the liver, liver only does a partial oxidation. For example, the glucose is only partially oxidized to lactate, right? And some other nutrients as well. So for the glucose, the liver only does the partial oxidation and to conserve the most of the carbon for gluconeogenesis, so make a glucose, right? So so that makes sense, right? The liver is a place that make a glucose. So glucose metabolized in the liver only goes partial oxidation and save the skeleton carbon for gluconeogenesis. And for fatty acid oxidation, we also only goes partial oxidation, meaning that fatty acids pick up from the circulation during the starvation, of course, right? And then the partial oxidized to acetyl-CoA, right? Fatty acids goes beta oxidation and generate a lot of acetyl-CoA, and this acetyl-CoA will not further oxidize in the TC cycle that stop at the acetyl-CoA, and acetyl-CoA then be utilized for synthesis of ketone bodies, right? So liver does a partial oxidation has its own reason, right? Because it's a place to make glucose and make ketone bodies, right? So again, couple of things that are specific for the liver. First is the primary energy fuse for the liver is the amino acids, alpha ketoglutarate, alpha keto acid, right? And second thing is that the liver partial oxidize and all the energy fuse, and the reason is for glucose neogenesis and for ketone body synthesis, right? The partial oxidized glucose and partial oxidized fatty acids. And the synthesis or release of energy fuse, you guys already probably known, right? First is glucose. So glucose is released from the liver, basically involve the two pathways, right? Glycogen breakdown and the gluconeogenesis, right? And it's also released ketone bodies, right, during starvation, and triglyceride. Triglyceride is released in the fat condition, right? Remember through the VLDL lipoproteins, right, transported all the triglyceride and to the other tissues. And just wanted to mention again that glucose ketone bodies are released in the from the liver in the fasted condition, right, fasted state, and triglyceride released in the basically in the fat condition. But we also mentioned, remember, triglyceride can also be released by VLDL during the starvation, right? And this triglyceride eventually being picked up by the muscle and oxidized in the muscle during the starvation, right? Okay, so this slide I just put all this pathway or fuel utilization pathways together in the liver, and we separate them like in the fat and fasting condition, how liver utilize different fuels and how liver metabolize the fuels and how liver made or contribute metabolic fuels to the body, right? So in the fat condition, we eat food and including carbohydrates, lipids, and amino acids, right? The proteins. So let's talk about the dietary glucose. We know that 20% of the circulating glucose initially from diet, right, picked up by liver. And the most important thing, the first thing that what liver will do with this glucose. In the fat condition, to make glycogen, right? So the first thing is to make glycogen. And this glucose, of course, can also be oxidized through the glycolysis pathway, which then generate pyruvate. And pyruvate can be further oxidized to acetyl-CoA, right? And here we're talking about again in the fat condition, right? So, and it can go to the TCA cycles, right, for complete oxidized. But again, in the liver, we know that the glucose basically will not complete oxidized or utilized for energy, right? If we have excess of glucose, and we can have a lot of acetyl-CoA produced, right? And this acetyl-CoA being basically used for fatty acid synthesis through the called a lipogenesis pathway, right? So converted glucose to the fat in the fat conditions, right? So this also is a way that save energy, right, in the liver. Will not complete oxidize, but converted it carbohydrate to fatty acids and stored in the adipose tissue in the fat condition, right? And fatty acids, of course, being re-acidified to triglyceride and packed to VLDL and released into the circulation in the fat condition will go to the adipose tissue for storage, right, in the fat condition. And second thing is that we also get a lot of fat, right, lipids from the diet. So what liver will do with the dietary fatty acids, remember, through the chylomicron remnant, right, then pick up by the by the liver. So liver also gets fatty acids from the dietary fat, and this will also be converted to triglyceride and in the VLDL release, and eventually also pick up by the adipose tissue. So this sort of the secondary pathway, for transport dietary lipid to the adipose tissue for storage in the fat condition, right? And then third thing is about amino acids, dietary proteins that we uptake, and a lot of fatty amino acids actually can be metabolized to pyruvate, right? And through this pathway, right? So either for lipogenesis, so again, we access protein. If we take up excess protein, it also be able to convert it to fat and stored in the body, right? Not just the glucose, right? So lipogenesis, that including the two precursors from glucose and the protein amino acids, right? And pyruvate can also be converted to the glucose 6-phosphate and for glycogen synthesis, right? So dietary amino acids can also be used for glycogen synthesis, right? So here are just the concept I wanted to remind you again. For glycogen synthesis, we not only converted glucose but also amino acids to glycogen, right? So in other words, access both glucose and amino acids can all be converted to glycogen and stored in the body in the fat condition, right? And also for the fatty acids, same thing, right? Excess glucose and amino acids can all be converted to fatty acids, right? See the pathway. And dietary, this is a one pathway, it depends on the type of amino acids. This type of amino acids can go this pathway, like converted pyruvate and glucose 6-phosphate and glycogen, right? And for the other set of the amino acids, glutamine, and the other histine, arginine, and this type of this set of the amino acids can also be used for gluconeogenesis and glycogen synthesis, right, in the fat condition, but through the different pathways. So this set of protein amino acids being converted alpha-ketoglutarate and succinyl-CoA and enter TC cycle to convert the oxaloacetate and enter the gluconeogenic pathway to generate glucose 6-phosphate and eventually convert it to glycogen, right? So and in the fat condition, if you still remember that gluconeogenesis pathway also active, right? And this pathway is a little bit specific, and they only use this set of amino acids as the precursors, right? And this new gluconeogenesis actually to make glucose 6-phosphate, and the purpose of this gluconeogenesis is not to release glucose into circulation, but to synthesize glycogen, right? Remember this, right? Any question about this slide? Right? Okay. And a fasting condition, right? The fasting condition, the liver, we know liver make glucose and make ketone bodies, right, and release into the circulation. So first of all, glycogen should be break down, right, to release glucose. And the second thing is the gluconeogenesis, right? And glycerol, lactate, amino acids, right, can all be pyruvate, can be all as the precursors for gluconeogenesis. And during the fasting, the glucose only partially oxidized to lactate, right? And lactate be saved. Lactate will not release from the liver, right? Because liver is the place that make glucose. So lactate not released, still stay inside of the liver, but can be used to convert the pyruvate and enter the gluconeogenic pathway for making glucose, right? So it's different from the muscle and red blood cells, right? They make lactate, but they release it because they are not the place that, you know, the making glucose, right? And of course, a small amount of glucose also be able to oxidize to acetyl-CoA and compete oxidize in TCA cycles. And fatty acids during the fasting, right? Fatty acids levels be very high because of lipolysis increase, right, in the adipose tissue. So liver picks up the fatty acids from lipolysis, and for oxidation, beta oxidation, and again we mentioned earlier, the fatty acids is also partially oxidized and stop at the acetyl-CoA, right? And acetyl-CoA is used for making ketone bodies, right, and release into the circulation fasting. And also some fatty acids from lipolysis being used to convert it back to triglyceride and make VLDL release into the circulation, and again during the fasting, and this VLDL primarily will go to the skeletal muscle, right, and skeletal muscle pick up the release the LDL, remember LDL, right, and hydrolyze triglyceride in VLDL and pick up fatty acids, and this fatty acids primarily oxidize for energy in the skeletal muscle during fasting condition, right? Right, that clear? Any questions? All right, so I think we just stop here and we have informal writing. Okay, so today's informal writing, so you guys please write something like, how does the skeletal muscle use the different fuels during feeding and fasting? Very simple, right?